Good morning. My name is Behjati. I'm from Sangha. I'll present some of the kidney work that's been undertaken around the globe. How would that work? All right, so I'll start by this. I just wanted to outline to you what the problem entails. So in trying to capture the different cell types of the kidney, what one needs to consider is that there are different anatomical regions in the kidney. So there's the, does this work? Oh, yeah. So there's the cortex, there's the medulla, and the pelvis, which is the collecting bit of the kidney, and in each bit there are different cells. And although let's say in cortex and medulla, some of the cell types are equivalent, there may be subtle nuances that one would have to capture in one way or the other. Above and beyond that, there's then the issue of the actual cell types. So in the nephron is the bit of the kidney that makes the urine up here, and then it gets sort of reabsorbed and modified as it goes through this system here, through this tubular system. And there are lots and lots of different cell types that one needs to capture in one way or the other. Now, the other complication in the kidney is that blood vessels are not just there to supply blood, they're intricately involved in forming urine. So the nephron, next to all these different tubules, sit different blood vessels, different vascular endothelia that specialize in interacting with the epithelium. That is the other thing that one needs to capture in these experiments. And then finally, there's, of course, also a whole bunch of immune cells that one would have to look at. So it's not just about the kidney epithelium, it's about the kidney epithelium in different anatomical spaces and the interaction with surrounding cells. And then finally, the bit that gets me excited are kidney diseases. So this is a kidney tumor, but above and beyond sort of tumors, there's a whole bunch of inflammatory conditions that I would have thought will really benefit from the Human Cell Atlas effort, in that we can move beyond this histological description, there is some inflammation, to a more quantitatively robust definition of what this inflammation actually is. And I think this is where the real merit lies. Although I would love to just talk about tumors, I think in the long term, this sort of the transcriptional data will be more informative for the non-oncological inflammatory conditions. So I'll show you some of the work that has been done. This is by no way complete, but really what I try to focus on are either published or pre-printed pieces of work that you can access and read for yourself. And surprisingly enough, there appears to be at the moment a slight bias towards developmental kidney work. So there's this lovely paper here, and how do we go back there? From Charles and Tang Labs, and what they did is they looked at human developmental kidneys, cataloged the cells, annotated them, and did some really quite interesting analysis which revealed this particular population of self-renewing cells within the developing kidney. So that might tell us, so the way kidney development works is that when we develop, the kidney bits form, and then when we are born, that process stops, and it might be that it's just this particular population here that is driving the formation of the kidney whilst we develop. Then there's this piece of work here from Sabrian Kretzler and Spence's lab, which gives a global perspective on all the different populations of the developing kidney, but then also does quite tremendous, it's a quite tremendous piece of work in looking into the different anatomical regions and asking actually what is the development within a glomerulus, within a particular tubular population, and so forth. And then there's this piece of work here from us, from myself, Mena, Maz, and Sarah. And we generated a census of normal kidneys from the developing kidneys, children, and also from adults. And the purpose of this census of cell types, as it were, was to provide a reference map so we can ask the question, what are kidney tumor cells? And we looked at two types of tumor cells. We looked at children's tumors, Wilms tumor, and adult clear cell renal cell carcinoma. And the sort of two interesting findings were as follows. For the children's kidney, what we found is we can correlate them with very specific developmental nephron populations. And for the adult tumors, and that was very surprising, irrespective of patient and cancer cell population and somatic genotype, when we look at what the cell type is, it turned out to be the same quite rare type of proximal tubular cells, which is surprising because obviously when we think about cancer, we think about heterogeneity and diversity, and how are we ever going to make any sense out of all the differences between patients, that maybe if we collapse all of that diversity onto cell type, that we can get around that problem and maybe perhaps consider the heterogeneity to be a distraction and just focus on the cell type. I thought I'd sort of provide you this tick list and to give you a sense of where we are. we clearly do have a census of both developing and mature kidneys, and as we've seen on Avi's slide earlier, there's much more data here coming from the Broad from Anna's lab. We sort of started to do the lifespan thing where we look at developing kidneys at children and at adults. We've had a crack at disease, but clearly what is required, a lot more mature kidney data to balance this discrepancy out between developing and mature kidney data as it is at the moment. I'd say there is some spatial resolution, but clearly we are far away from a comprehensive atlas. And then I think we need to have a real go at the non-malignant conditions, which I think will be hugely informative and transformative for kidney disease in the long term. Thank you.